Hello, everyone. This is Muhan from Johns Hopkins University. Today, my topic is reconstruction and the refinement of crossing muscle fibers in the human tongue. The tongue plays an important role in multiple basic human functions, including breathing, swallowing, and speaking. These functions are possible because the tongue can produce complex deformations with its muscle architecture. This figure shows a sagittal slice of a T2-weighted magnetic resonance imaging of a human tongue. The yellow square is a bounding box of the tongue region. The human tongue is made entirely of muscle fibers that are extensively interdigitated and orthogonal in 3 dimensions. Some regions contain a single fiber orientation, but most regions are interdigitated with 2 fiber orientations crossing at approximately 90 degrees. These 2 figures show mid-sagittal slice of idealized tongue muscle fiber orientations from a mechanical tongue model. In the figure, the first fiber image mainly contains the genioglossus muscle and has fan-shaped appearance. And the second fiber image contains superior longitudinal and transversus muscle. Most of the transversus muscle have left-right orientation, which is perpendicular to the screen. There are many scientific reasons to obtain a better understanding of the relationship between individual tongue muscles and the complex functions. For example, this can help us better predict the functional outcome after surgical resection of the tongue. Diffusion-weighted imaging has been used as a non-invasive method to image tongue muscular architecture. DWI can quantify the self-diffusion of water in biological tissues. And this is done by measuring the attenuation of the MRI signal in the presence of diffusion encoding gradients. High angular resolution diffusion imaging technique, which is one imaging type of DWI, can be used to discriminate crossing fibers within the same voxel. Currently, most existing fiber reconstructions methods from Hardy were designed for brain tissue and do not account for the orthogonal nature of the crossing muscle fibers in the tongue. We previously proposed a convolutional neural network that can reconstruct both the fiber orientation distribution function and the fiber orientations from Hardy tongue data in a fully automatic manner. We introduced a separation loss during training to encourage orthogonality of the predicted crossing fibers, which is important in tongue muscle fiber reconstruction. The fiber reconstruction network outputs 2 fiber images. The network estimates the primary orientation based on the signal fractions, and the primary orientation is intended to be more dominant in that voxel. However, the assignment of the first and the second fiber orientations is not always correct, which means the assignment might disagree with a neighboring voxel. Here is an example of fiber reconstruction results from an algorithm. In most regions, the genioglossus muscle appears as the first fiber orientation, and the transversus muscle is the second fiber orientation. However, these yellow contours point to some regions where the fiber assignments are not correct, so we get inconsistent visualization results. In this paper, we proposed an automatic fiber meshing algorithm to refine the assignments of the reconstructed fiber orientations from our convolutional neural network. We formed an optimization framework to find an optimal assignment for each voxel. The proposed method provided better fiber assignments on both synthetic tongue data with different noise levels and on data from a post-mortem human tongue. And this method can also be used as a post-processing step for other fiber reconstruction algorithms. Here shows an example. The fiber matching method improves the visualization of the fiber orientations for further analysis. And this example shows that the fiber orientations after matching are more consistent. Here, I will briefly talk about our previously proposed fiber reconstruction network. The network architecture is composed of two stages. The input to the network is defined as the spherical harmonic coefficients of the Hardy signal. The stage 1, which is in the solid green box, predicts the spherical harmonic coefficients of the fiber orientation distribution function. And the stage 2, which is in the dots and dashes green box, predicts two binary terms indicating if the first and second fibers exist, and two vectors, D1 and D2, representing the fiber orientations, which simulates the peak finding process in fiber reconstruction methods from Hardy. The loss function is a combination of mean squared error and fiber orientation error. We also introduced the separation loss to encourage the orthogonality of the crossing muscle fibers. And this separation loss is defined as the absolute value of the cosine similarity of the estimated crossing fibers. This separation loss at each voxel will be minimized when the crossing angle of the two predicted orientations is 90 degrees. Therefore, the network is encouraged to predict orthogonal crossing fibers, which is an important characteristic in tongue muscles. The fiber reconstruction network predicts up to two unit vectors, D1 and D2 per voxel, that represent the fiber orientations. And these D1 and D2 will be the input to our fiber matching algorithm. When there are two crossing fibers, the one that is assigned to be the primary one is intended to be more dominant in that voxel, and we refer to as D1. When there is only one fiber, it is always assigned to D1, and D2 is a zero vector. However, the assignment in one voxel might disagree with a neighboring voxel. Given a pair of vector images that represent the fiber orientations generated by the fiber reconstruction method, our goal is to switch the assignments between D1 and D2, or reassign D1 to D2 in the voxels with incorrect assignments. We consider two neighboring voxels, P and Q, and the two orientations at each voxel, D1P, D2P, D1Q, and D2Q. We define the following quantities that measure the similarity between orientations in voxels P and Q under different assignments. Aij is the absolute value of the cosine similarity between the ith orientation in voxel P and the jth orientation in voxel Q. Let's look at an example of the similarity measurements in ideal case. Suppose we have neighboring voxels P and Q. The blue arrows represent the first orientations, and the orange arrows represent the second orientations. In this ideal case, we assume that neighboring voxels P and Q have the same fiber orientations, and D1 and D2 are orthogonal in each voxel. In the case where the assignments are correct, A11 is the inner product of the two blue vectors, which is 1, and A12 is the inner product of the blue vector in P and the orange vector in Q, which is 0, and the same for A21 and A22. So in the correct assignment case, A11 and A22 are large. Then let's look at a case with incorrect assignment. We see that the assignment of D1 and D2 in voxel P or Q should be switched. In this case, A11 is still the inner product of the two blue vectors, but is 0 in this case. And A12 is 1, and similar to A21 and A22. So in the incorrect assignment case, A12 and A21 are large. We introduce a binary variable y in every voxel to indicate if the current assignment is correct. If y is 1, we will keep the current assignment. If y is 0, we will switch the assignment of D1 and D2. We assume the fiber orientations after matching are D1 star and D2 star, which can be written as equations of y and D1 and D2. Our objective function for all the voxels in the image is formed as this equation, where the script E is the set of all neighboring voxel pairs in the image. This objective function is designed to maximize the similarity between the first orientations in neighboring voxels and the second orientations in neighboring voxels, which makes the display of fiber orientations more consistent. The objective function can be expanded as a function of y and aij, and it can be simplified as this equation, which ignores the constant term. This optimization can be modeled as a quadratic unconstrained binary optimization problem, which is an unconstrained model other than the binary restrictions on the decision variables yp and yq. Solving this Kubo model gives y in each voxel to determine if the assignment of D1 and D2 should be switched. We quantitatively evaluated our fiber matching algorithm on synthetic tongue muscle fibers with manually created incorrect assignments. The original ground truth fiber orientations were generated based on a mechanical tongue model. There were more than 10,000 voxels containing fibers, and each voxel consists of at most 2 crossing fibers. To generate our testing images, there are 2 steps. The first step is to rotate the fibers in each voxel along a random axis by a small random degree alpha, and this alpha follows a uniform distribution. The second step is to randomly select R% of the voxels and switch the fiber assignments in that voxel to simulate the output from the fiber reconstruction method. In our experiment, theta was set to 10, 20, and 30 degrees, and the ratio of the switched voxel R was set to 10, 20 until 90. For each combination of theta and R, 2 pairs of fiber orientation images were generated with independent realizations. Here is a visualization result of the fiber matching on the synthetic tongue data. In this experiment, R is 40 and theta is 30 degrees. Before matching, there are many inconsistent assignments. And after matching, we can see that the first orientation image contains the genioglossus muscle, and the second contains the superior longitudinal and the transversus muscles. It is worth noting that our objective function does not enforce the first orientation to be the first after correction. Our goal is to create consistent fiber orientation images. Therefore, when R is greater than 50, the algorithm is likely to generate completely inverse first and second orientation images. Here is an example. In this case, R is 70 and theta is 20 degrees. The algorithm produces completely opposite first and second fiber images. The first orientation contains superior longitudinal and transversus, and the second contains genioglossus. But still, the fiber orientations are more consistent after correction. The performance of the proposed method was quantitatively evaluated using the assignment error rate, which is defined as the number of voxels with incorrect assignment divided by the total number of voxels that contain fibers. This table presents the assignment error rate of the fiber matching algorithm on the synthetic fiber images. We can see that the proposed method greatly reduces the number of voxels that have incorrect assignments. For example, when R is 40 and theta is 30 degrees as pointed by the green circle, the error rate before fiber matching is 40% and it reduced to 0.248%. We applied the fiber reconstruction network and the fiber matching algorithm on data from a post-mortem human tongue. The algorithm only processed the tongue region restricted by a manually delineated tongue mask. And this figure shows a visual result of the fiber orientation reconstruction results produced by the network. We see that the fiber reconstruction network can resolve crossing muscle fiber in the tongue and predict orthogonal fiber orientations. However, we can also observe that in some regions where the transversus and genioglossus muscles are crossing, see the yellow contours, the transversus muscle fibers appear as the first orientation, which is not consistent with most other regions of the transversus muscle. The reason for this inconsistency could be that the strength of the transversus muscle is higher than other muscles in those regions. After fiber matching, we see that the fiber matching algorithm switched the fiber assignments in those regions and provided cleaner fiber orientations. The proposed fiber matching algorithm can also be used as a post-processing step for other fiber reconstruction methods. This figure shows the fiber predictions on the same post-mortem tongue produced by a widely used fiber reconstruction algorithm, the constrained spherical deconvolution method. The yellow contours point to some inconsistent assignments, and these regions were corrected by the fiber matching algorithm. In this work, we proposed a fiber matching algorithm that can refine the assignments of the tongue fiber orientations reconstructed from Hardy data. We formed a quadratic unconstrained binary optimization model to maximize the similarity between the first or second fibers in neighboring voxels. The proposed fiber matching algorithm can also be used as a post-processing step for other fiber reconstruction methods. Our method improves the visualization of the fiber orientations for further analysis on crossing muscle fibers. That's all. Thank you. Thank you for your listening.